me right now. Now I would like to hand over to Professor Dr. Gert Kema from Wageningen University and Research, and he will be talking today about the ongoing pandemic of tropical race 4 threatens global banana production. Thank you very much, Gert, for joining us today, and we are looking forward to your talk.
Well, thank you very much. Thank you for hosting this talk. I'm ready to go, so I will switch off my camera and then we kick it off. So there we are. So the title of the talk is The Ongoing Pandemic of Tropical Race 4 that Threatens Global Banana Production. Of course, we are in a time of pandemics and one which is much more serious than TR4 and banana. But nevertheless, I will also make a link to the current COVID-19 pandemic later on during the talk as well. So my talk will revolve around why are we in this state, how did it happen, and what are we up to next. So I start with this painting of Pieter Bruegel de Oude, which is called The Harvest, painted in 1565. And where I would like to draw your attention to is the height of the crop. Excuse me. And so as you can see, the height of the crop is about the height of the people working in that wheat plot in 1565. So what we have to keep in mind is that, of course, the average height of people has increased over time. As you can see here in this slide, over 2,000 years, but even still in the early 1800s, people were about 166 or so, and only over the last 200 years, our height has increased significantly, primarily due to improved nutrition. And if you look then to the height of a wheat crop, as you can see here, so old wheat crops used to be very tall, but they have decreased in height over time significantly, all driven by gene discovery. And so using particular genes, the RST genes, introducing them, reduced the overall height of wheat crops, and that of course contributed also to the overall yield increase, because the risk of lodging was reduced. You can put more fertilizers, which is boosting yield, of course, along with genetic improvement through breeding. So this was one of the factors contributing to the Green Revolution, for which Norman Borlaug got a Nobel Peace Prize. And of course, he also bred for daylight insensitivity. So his wheats could be grown over various different environmental zones. So I come back to wheat a little bit more. Many of you may know that I worked on wheat for more than 25 years and only over the last 10 years kind of swapped to bananas. So I always can make this comparison to where we are in wheat R&D compared to banana, which is interesting. So this is how a contemporary wheat field looks like. So this is in northern Tunisia, Bejá. This is a durum wheat plot. And so the overall size of this wheat crop is about 50 to max 60 centimeters. So that is contemporary wheat varieties are very diverse, number one. There's an immense amount of breeding effort, and the overall length of the crop has reduced significantly. Now we switch to banana. So first of all, what I really would like to underscore is the importance of banana as a food crop. Now, many of us may know banana as a dessert fruit, but as you can see here from the slide, banana is a very important food source for millions of people here in Southeast Asia. Africa is a true staple in East Africa, primarily cooking bananas or East African highland bananas, and of course Latin America. And so most of the bananas that are being exported to Europe, for instance, are produced in Latin America. Most of the bananas exported here, for instance, to Japan and Korea are all or primarily produced in the Philippines. Now, if you look at the global, and of course, what we need to underscore is that in this entire banana business are millions of people involved, primarily smallholders, but it also provides a lot of employment for millions of workers in plantations, because banana is a very intensive crop, requires a lot of hand labor. Now, if you look to the overall picture, it is stunning to see that Cavendish varieties, and those are the varieties we usually buy in Western supermarkets, really dominate the trade. So 95% of the export bananas is Cavendish. But if you look at a global level, and that's what you see here in the pie chart, 50% of global production is also Cavendish. So Cavendish is not only important for the export, but it's also very important for major domestic markets. For instance, 50% of the Indian market is Cavendish bananas as well. China does not export bananas, or barely, 100% Cavendish, etc. So this is very important. And of course, the question is, why is that? Why do we have such a massive monoculture of Cavendish bananas varieties around the world? And that is this reason, driven by a plant disease that's called Fusarium wilt, caused by a suite of Fusarium fungi that wiped out the Gros Michel production in the previous century in Central America. If you Google it, you will find it that Gros Michel was really knocked down, caused a huge unemployment in the area. And what you see here in the slide was one way to deal with the disease, and that is inundation. So by inundation, you essentially flood the land, you generate an anaerobic environment. It's a soil-borne fungi. These are soil-borne fungi, so you suffocate the fungus essentially, and that kills it. And then the same plot was replanted, as you can see here. So this is Gros Michel. In those days, there was no tissue culture, so all plants were produced by suckers. Those suckers were then planted in the field, but in many cases, unfortunately, those suckers were infected, but you just couldn't see it yet. And of course, then you plant infected suckers in an area that has been inundated, where there is no biocontrol, there is no competing fungi or bacteria in the soil. So it's a kind of virgin land planted with infected suckers, and that of course boomed the epidemic. And that resulted then in really shifting cultivation. So at a point of time, the United Food Company, for instance, owned about 50% of the arable land in Guatemala. Now, eventually, of course, this could not go on. And therefore, new varieties had to be used to replace Gros Michel production. And that was Cavendish. And that really boomed the Cavendish production. So what you see here is a Cavendish area in northern Colombia, in Urabá, 40,000 hectares, one single set of varieties, Cavendish. So why was Cavendish so popular? Cavendish is not a product of a breeding program. It's essentially a wild clone that was collected by botanists, survived in several botanical gardens, ended up in the UK, in Derbyshire, and there it was found later on, as it was given to reverends and missionaries that took it to other areas, and there it was found by accident that it is resistant to race 1 strain that caused the epidemic in Gros Michel. So of course initially there was reluctance to embrace Cavendish because it came with a couple of additional requirements, for instance for the logistics of the fruit. So in Cavendish bananas you have to pack the fruit into boxes, you have to cool them and then transport them and ripen them on the spot. In Gros Michel that was really different. There you could harvest by the bunch, you throw the bunch in a train or in a ship and transport it, for instance, to the US market. And so the entire logistic chain for Cavendish had to be tailored. That came also with an immense investment. So that makes also the production and the swap to other cultivars very inflexible. But nevertheless, Cavendish succeeded because of its extreme high level of resistance to race 1 strains that hold until now. And so this is a fantastic example of durable resistance that holds already for close to 80 years, despite that Cavendish is grown throughout the world in soil that have totally been infested with race 1 strains. I know plots in Australia, and Andre Drent, my colleague, showed them to me while I was there, but also in Costa Rica, where you plant now Gros Michel in those infested plots. Within 9 months, those plants are infected. And so the soil is still contaminated. Cavendish is excelling, super resistant to the strains that caused the previous Fusarium wilt epidemic. So this is the reason why Cavendish dominates the market. But it comes with a couple of complexities. And one of them is this disease, black Sigatoka, caused by Pseudocercospora fijiensis. very much related to Zymosatora tritici in wheat, the fungus where I worked on for many years. Both Dothyriomycetes, very sexually active fungus. Each and every isolate is a genetic, unique individual, airborne, spread around the world. And the only way to control the disease is by frequent fungicide application. That's what you see here in Costa Rica. This helicopter is flying on this particular plantation, the Cartagena plantation, more than 60 times per year, close to 70. Every 5 days it has to return to control black Sigatoka because Cavendish is so super susceptible. All right. So if you move north of Costa Rica or south, the number of applications is reducing maybe to 35, 40 times. And that is simply driven by environmental conditions. And so up to the level that you can grow Cavendish into the desert high plains in Peru, where organic bananas are being produced because there is no black Sigatoka there. And so Sigatoka requires an immense chemical input to control the disease and make sure that we have those bananas in the shelf in the supermarkets. The second disease is Fusarium wilt again. Here you see a plot in the Philippines that is infested by TR4, the topic of our talk today. Tropical race 4. We gave it a new Fusarium species name. We rearranged the taxonomy. It was previously known as Fusarium oxysprum cubensis, so Oxysprum formispecialis cubensis, infecting bananas. I come back to that later, but here you see that TR4 is really crashing Cavendish production, not only in the Philippines, but in many, many other areas. So we're going to explore this a little bit more in the forthcoming slides. So remember, Cavendish very resistant to race 1 strains, very susceptible to TR4. And what we have to keep in mind, and I already put in now, TR4 is not a mutant of a race 1 strain. TR4 is a separate genetic lineage. And so it's a single species that is currently disseminating and killing Cavendish fields throughout the world. So this is Fusarium wilt. So here you see the symptom of an infected plant. You see the chlorosis of the leaves here. It's a soil-borne fungus. It enters the plant through the roots. Then it's colonized in the vasculature and eventually occludes the xylem, which is resulting in this chlorosis, and eventually it kills the plant. So as I said, in Gros Michel, Fusarium wilt was called by a plethora of clonal Fusarium species. In Cavendish, Fusarium wilt is called by a single species called, we call it tropical race 4. Now what is also important, and I already mentioned that, is very persistent in the soil. So the fungus can survive many years as chlamydospores in the soil, but it also easily enters other hosts as an endophyte. So this combination makes the disease very persistent, which essentially means that you cannot return with the same variety even after multiple years. So of course, if you would have different varieties, you just place it, and Cavendish is the prime example. These soils are infested with race 1 strains, nevertheless you can grow Cavendish. But at this stage we don't have anything that can replace Cavendish that has adequate levels of resistance. And of course the example of the level of resistance we're looking for is the resistance that Cavendish has to the race 1 strains, because there we have the practical example of durability for many decades. So this of course is raising questions on the entire epidemiology of the fungus. So despite the selection pressure you put on the population by growing Cavendish varieties, not a single mutant of race 1 isolates has been identified that are able to overcome the resistance in Cavendish. And so this is the kind of the framework we're talking about. So this is the internal symptom if you cut a banana plant. So you see the occlusion of the veins here. So this is all rotting, this is full of inoculum, but here the yellow spot is exactly the same. So a pseudostem of a banana plant is a massive source of inoculum. Once you cut that plant, the aerosols that are being spread are full of spores. So that really requires special measures to manage the disease once it is entering a field. So just to highlight a little bit better or in more detail the threat of TR4 to banana production. So we go into a helicopter ride at this part of the presentation, and we're flying over the island of Mindanao in the southern Philippines, which is the top zone for Cavendish production. So 80,000 hectares roughly, all destined for export to Southeast Asia and the Middle East. And what you see here in those empty spots, those are all areas that are affected by TR4 in Cavendish plantations. I come back to this here, what you see here, this is rice hull burning, but you see here this river is going here, and that is flooding frequently, and flooding is a fantastic way for the fungus to disseminate. It is a soil-borne, so it is not, it's not spread by air or by wind, but primarily by infected plants and flooding, and of course, that does not explain the international dissemination, and there the major factor is man, bringing infected plants or contaminated tools, boots, soil. That's all the way how it's disseminating internationally. So if you now look out of the window of the helicopter, you see this vast area. This is an area of close to 50,000 hectares in the southern Philippines, one contiguous region of Cavendish banana production. And what you have to keep in mind is that these areas are surrounded by many, many smallholders. And those smallholders have usually limited awareness. We ran projects in the Philippines where we have really held questionnaires to identify what is the general know-how of the smallholders on recognizing the disease, and that was very, very limited. And that's why we also see many artisanal solutions to deal with the disease, and therefore the efficacy of any measures, control measures, is usually small. I mention here the Chinese market because the Chinese market has a very big impact in the region, because China is really immensely suffering from tropical race 4, and therefore the spot market prices of bananas are increasing continuously, and the sourcing of bananas by the Chinese market is immense, up to the level that also plantations are really moved into the greater Mekong area just to make sure that there is enough bananas produced to satisfy the Chinese market. And so that is also important to keep in mind. So this is one of those artisanal ways to control the disease, and this is called rice hull burning. So what you see here is what these people do in the plantation. This is an infectant plant. They've chopped it down completely, cover it with rice hull. In the back you see another infected plant here. So this one here is also infected by TR4, but they selected this one, chop it down entirely, cover it with rice hull, and then set the rice hull on fire. And that starts smoldering for a couple of days, or maybe even up to a week or a little longer. And the general understanding and belief is that this is controlling the disease. And so you reduce or you kill the inoculum. This is what you see then. So it's being set on fire, smoldering. This is common practice in the Philippines. It is very costly because of the limited or more and more limited access to rice hull. And of course, what we really discussed when we started this research is to see, what is now the efficacy of this measure. And that was part of the PhD thesis of one of my students, Maricar Salasina. So this is just a slide from her thesis that will be published soon. And what you see here in the top panel is we sampled 25 centimeters, 50, 75, and 1 meter. And here you see the DNA quantification of TR4 prior and after burning. And as you can see, by burning, even at 25 centimeters, although I was surprised by the temperature, the temperature can really be 300 degrees or so, but you do not eradicate the fungus. And that is important to keep in mind. So you walk around the entire area, and by walking you have the infested soil under your shoes, and of course this also actually contributes to the epidemiology of the fungus. So this is now DNA. Here we had samples buried in the soil, where we really looked at the viability of the recovered Glamida spores. And here you can see that at 10, 20, and 40, we were not able to recover colonies from those samples that were buried either after rice hull burning or wood burning. but at 1 meter we still recover levels of chlamydospores as in the controls. And so it really shows you that burning is costly, it's time intensive, but it does not eradicate the fungus, and the fungus really can survive in rather deep layers of the soil. And so this is not a sustaining management option. Nevertheless, practiced broadly throughout the Philippines. So, and that brings me to this slide. So if you would read a book of John Saluri, which is on the Honduran banana production around the time that Gros Michel was produced and the demise of Gros Michel in that entire area, you see that many options were tried to battle and to really conquer Fusarium wilt in Gros Michel. And to put it very briefly, at the end of the day, nothing worked. But this is, of course, a century ago, and unfortunately, many of us around us have forgotten the lessons that we can learn from what was done during the Gros Michel time. So then I read this Nature briefing last Tuesday, which was on the COVID-19 epidemic. But you see a very similar thing happening. You know, when TR4 enters a country, the country is swamped by people that have solutions, snake oil doctors that can sell their products to deal with TR4. And here in the COVID-19 epidemic, you see something similar, which made Timothy Caulfield saying, you know, the scientific community must take up cudgels in the battle against bunk. We must stop legitimizing pseudoscience. And the best way to fight misinformation is, let's get swamping. Now, if you look here at some statistics I looked up, if you look at the Bob McIntosh's list of gene symbols in wheat, you see that the number of genes discovered in wheat and described is roughly 8,500. And more than thousands of those genes are related to plant diseases and pests. And many of these have been validated and mapped. If you compare that where we are in banana genetics, I think we know less than 25 genes, despite we have the genome, and I'm talking about validated genes. But for disease resistance, we have essentially only 2 genes identified. The first one was done by James Dale's group, who cloned a resistance gene from Musa acuminata malacensis, which is the wild A-genome donor of the current edible bananas. So he cloned that gene, put it in Cavendish, and showed that yes, that's a single gene, and it provides a level of resistance which is complete, as we see very similar to Cavendish to raise 1. You see a transformed Cavendish with that gene has an adequate level of resistance to TR4. The second gene that's being discovered is an R program that's mapped and that deals with resistance to raise 1. For black Sigatoka, despite its importance, not a single resistance gene has been identified. And of course, the question is, why is that? Now, wheat is a crop that you can cross, and the yield is the seed, right? Well, but in banana, we don't want to have the seeds. While bananas are full of seeds, we want to have seedless fruits in order to eat them. So that complicates breeding. On top of that, edible bananas are mostly triploids. They're sterile. So if you want to breed bananas, you have to go back to the wild diploids and start from there. And that, of course, complicates it. It's definitely a complex crop to breed. But if you talk about swamping, together with a group of colleagues, we started the Frontiers in Plant Science research topic on Fusarium wilt. And as you can see, up to now, we have about, let's say, almost 100,000 views. There are multiple articles there. And so by generating data, accurate data, and my goal is really to focus very, very much on genetics, because if we want to improve banana, if we want to deploy the genetic resources that are available, we have to do much, much more genetics. And so there is considerable attention for the work on banana nowadays, which is great. So we can increase the community, and that will, of course, increase also the quality and the overall output. Okay, so where we got started in a collaborative project with Indonesia, one of my students, Fajar Ahmad, for instance, he traveled and crossed entire Sumatra and mapped and identified and described wild diploids across the island. And so this is a fantastic resource that we can eventually use to discover genes and to deploy them. Earlier on, Cavendish, very resistant to RACE1, very susceptible to TR4, and Gros Michel, very susceptible to both. One of the questions we raised was, despite the fact that Cavendish is resistant already for decades to RACE1 strains that are disseminated around the globe, why do we not yet know which genes actually are involved in protecting Cavendish to RACE1 strains? Now, I already mentioned one of the complexities is that Cavendish is a triploid, so you cannot readily make a segregating population and do genetics. So what Bejá did, he made a cross of Musa acuminata malacensis, the wild diploid donor, and made a biparental mapping population and started screening that. And in a 4-year program, we were able to identify a major QTL on chromosome 10. And so we are currently narrowing down the QTL. There are several candidates there. Of course, we don't know whether it's a single gene, yes or no, but at least we have identified the QTL that is responsible for resistance to RACE1. So despite banana is a complex crop, at a diploid level, you can make big, massive populations, genotype them, phenotype them, and that is the way forward for gene discovery. So we are very hopeful this can be published this year, which is then the first gene identified, or the first locus identified for RACE1 resistance. And when we started banana research about 10 years ago, my goal really was, if we want to have an impact, TR4 was at those days not yet discovered outside Southeast Asia. Let's make a molecular diagnostic which is really shortening the time to diagnose the disease, so that we can rapidly, within 1 day say, hey, this is TR4, and we don't have to wait for several months after a vegetative compatibility test. So this was a breakthrough, was a nice paper, mostly done by Michel Dita when he was in my lab as a postdoc. And that diagnostic has been used throughout the time that TR4 was spreading. So you see a lot of blinking things, and I did that just to draw your attention. So here you see 2014 blinking. This was the first time when we discovered TR4 outside Southeast Asia. And that kind of rocked the community, said, hey, TR4 is not anymore a Southeast Asian problem, it's on the move. And since that time, you see all those different dots. It has been discovered in multiple other countries, up to 2019, last year in Colombia, and even this year here in Mayotte and in Turkey. So in the far majority of cases, that diagnostic has been used to confirm, yes, this is TR4. So we also have very weird incursions that we really were surprised by, and that is, and so I already mentioned this, so the kit was used throughout these identification rounds. And why I'm showing this is because we published the primers that are diagnostic for TR4, but what time has shown that people start to experiment with the primers or the PCR conditions, which results in false positives or false negatives. So that's why we decided to work with a company that commercialized those primers into a fixed kit where you always follow exactly the same protocol. So this is an advantage for data exchange and also for precision, right? So everybody is using the same kit and you can compare data. So that was the main driver to make it commercially available so that also everybody also can buy it. Now, this is one of the weird incursions we have encountered. We were not involved in diagnosing TR4 here, but this is TR4 in the UK, surprisingly, last year. And this is called the Eden Project. It's in southern Cornwall. It's essentially a big greenhouse complex with many tropical crops, including banana. And even 10 years ago, they already saw, hey, those banana plants show symptoms and show yellowing, took them out, replaced them. And only recently they realized, wait a second, maybe this is due to a disease. And yes, they isolated the fungus from the plant, sequenced it, and you know what, TR4 in a greenhouse in the UK. And this really underscores how easy it is to disseminate this disease internationally. So we're talking about a greenhouse complex in a non-banana producing country, yet TR4 entered that specific location. Okay, so we move on from there. So in that same collaborative project with Indonesia, Nani Mariani was really zooming into Indonesia. And she sampled across the entire archipelago. You see the red dots, and collected a collection of more than 200 islets. And to our surprise, 50% of that entire collection was TR4. But what we also saw is that by doing the phylogenetics, you see here the different clusters that she identified, and what we showed is that the race 1 strains here in cluster 2 is not a single species, but essentially is multiple species. So you really see that in race 1 we're talking about different lineages. And so developing, for instance, a diagnostic for race 1 is not that easy because it's not clonal. These are different species. Whereas here you see that TR4 is a single genetic lineage. Now, of course, we wondered, why do we have TR4 disseminated across Indonesia, from here in Papua to the tip of Sumatra here in Aceh? And that was really most likely driven by the transmigration policy of the Dutch colonizers that started 150 years ago, but was only ended in 2015 by President Widodo. And so Nani also mentioned to me that she found TR4 only in those transmigration zones. So these are zones where people from Java moved there, started their own households, and there is where she found TR4, but not outside those zones. So most likely the movement of people across Indonesia contributed also to the dissemination of TR4 across the country. So I already highlighted the different species in the race 1 complex. And then with Nadia Ordóñez and Fernando, we continued with a global survey. So we, together with many partners, including Michel that I already mentioned, Andre Drenz and Randy Platz, have been very, very instrumental to help us to build that collection, but also many other national collaborators that I really would like to acknowledge here as well. We built a collection, and here you see the summary of an analysis of 623 islets across the globe. So all those islets have been genotyped, and many have been sequenced, up to about 65 islets that are sequenced. And that helps us to build the phylogeny of, let's say, the Fusarium complex that is causing Fusarium wilt in banana. And that's what David Torres did. He's a PhD student of Bartoma and Michael Zeidel and myself. And here you see the phylogenetic tree that he has built. So let's zoom in a little bit into that tree. So here you see the race 1 strains that I mentioned. And so you clearly can see this is not a single clade. So this is based on thousands of dart markers. So you really see here we have individual lineages underscoring what I just said, right? We have different species. So we took the consequence from the fact that Fusarium oxysporum is known to be polyphyletic, and the subspecies, or the Forma specialis cubensis, also polyphyletic. And so we took that consequence and said, okay, you know, these are actually different species. And of course, Nani did also all the required morphological work to underscore these genetic indications. Now, if you then look here in TR4, you see an entirely different picture. So you see that all the TR4 strains that we have genotyped and that we have sequenced essentially sit in one and the same clade. Yet there is a very small group here of Fusarium odoratissimum strains that are not TR4. And so there is some diversity, but the far majority in this cluster is TR4. So that brings us to this conclusion that race 1 is a complex of diverse species, and TR4 is closely grouped, and you see it here in the PCA, in a different genetic lineages. And TR4 is now distributed globally. And yet, while we still say that, yes, TR4 is a clone, and that's what you see here, right? So here you have the strains that we have sequenced in this paper. So this was on the Greater Mekong area. So we sampled in Vietnam, in Laos and Myanmar, and in China. You see, if you sequence them, they're all the same. They're very similar to the ones that we sequenced previously. Yet Michael built a bioinformatics pipeline, and Harold was also involved, where we use SNPs to see can we do any forensics and can we predict what is the origin of incursions. And what you can see here is that an incursion of TR4 that we discovered in Pakistan is clearly linked to a strain from the Philippines. Here you see also Laos, Vietnam and Myanmar is linked to China. And I already mentioned that China's banana producers are moving south because they don't have land anymore, because it's all infested with TR4. So they move to new areas in Myanmar, Laos and Vietnam, start plantations there, but eventually drag the disease in those zones as well. And that is really illustrated in this plot. The same we saw for Lebanon and for Jordan, of course very close, but those two islands also closely overlap. And so you can see, despite the fact we're talking about a clonal species, based on the sequencing we did and the SNPs that have been discovered throughout that pipeline, we can at least try to predict some forensic relationships. And that brings us in general to this conclusion. If you look at the diversity that we have discovered by genotyping this Fusarium panel of isolates, it actually kind of overlaps with the origin and dissemination of banana. So Southeast Asia is the origin of banana. Here you find the greatest diversity. For Fusarium is exactly the same way. If you move out of the center of diversity, the complexity of the populations is reduced, although we may have a second center of diversity here in Latin America. And so this bananas have been disseminated, as you can see, starting from the 200s or so up to the 1800s and later. And by shipping or taking banana plants along, many of those banana plants, of course, were infected. The disease has also been dragged to those new areas. Nowadays, shipping plants or taking plants, of course, is prohibited. So the most recent incursions of TR4 are most likely not linked to transporting infected plants, but by transporting infected or contaminated tools or infested soil. So I already mentioned the kit, right? And that has fulfilled the role for 10 years, but of course, a diagnostic is valuable until you run into an isolate that provides you with a false positive or a false negative. And this is something we expected to happen somehow, somewhere in the future. And that's why we have also started to work on a second diagnostic on a different genomic spot in the genome of Fusarium. And since we had all those sequences, we could really align all those sequences and identify what are other unique regions in the TR4 genome that we can use for additional diagnostics. So we developed a LAMP diagnostic. This requires less training, is cheaper, and you don't need a full lab. Of course, eventually, in my view, we have to go to this type of diagnostics that are really, high throughput, low cost, and can be used with anybody. Now, there were two papers that indeed discovered a false positive or a false negative of the first diagnostic, published last year by Freddy Magdama in Ecuador and by our colleagues in Australia from Andre Drent's group. And fortunately, since we had the LAMP diagnostic developed and also published that the same year, we could show that those false positives were fortunately correctly diagnosed by the LAMP diagnostic. So now we have two diagnostics in place, and the beauty is if diagnostic 1 is positive, diagnostic 2 should be positive too. And here you see a cartoon of the TR4 genome. This is the location for the first diagnostic we developed with Michel, and the second one is here on a unique genomic position on chromosome 11 that was used for LAMP. So we have now additional tools, and of course we have to continue to develop diagnostics to support the industry, growers and governments as much as we can. Now, we use those diagnostics also after the incursion of TR4 into Colombia. And of course this was a dramatic situation, because this was the first encounter of Latin America by TR4. We sequenced those strains again, so 3 strains from Guajira in Colombia, put them in the framework of the others that we sequenced, and you see these are all confirming again that we're talking about a TR4 unique genetic lineage. And that's why I come towards the finishing of my talk. But what do we need to do? Well, we need a disruptive change in prevention. So one of the scenarios we're working on is aerial monitoring. Mounting cameras under planes and scanning entire areas. We're not talking about a single farm. We're talking about thousands of hectares that we can monitor and identify risk zones. If we would have used this technology in Colombia ahead of time, I'm sure we would have diagnosed that incursion in Colombia way, way earlier than it was discovered now, because most likely Tier 4 was already in Colombia for at least a number of months, if not longer. It was an organic farm, it was confused with another disease, but from the air you will see those discolored plants right away. You can go to that spot, sample it, test it, check it, Tier 4, yes or no, if it is, isolate, etc. Now it took us, and particularly the Colombian partners, by surprise. And so with this technology, you really scan crops, as you can see. And so you can do pattern recognition. This all goes to a mobile app. You measure gravitation. So by using this technology, which is high throughput, you can really identify zones that are risky and go there and sample them. And so you don't wait until it surprises you. You go there ahead of time, proactively, and sample it. Secondly, we need a disruptive change in breeding. Very important, because the genetics is the source and the promise for the future. And we are way back, as I showed you, compared to many, many other crops. But also, it's of course also a fantastic opportunity. And if you think about breeding, we are not just talking about Fusarium, wilt, or Panama disease. We of course also talk about black Sigatoka, but in breeding we can address many, many other issues that we would like to improve. It can be quality, it can be shelf life, it can be the height of the crop. But as you can see, the overall cost of Sigatoka control, on average, in the areas where Sigatoka is very intense, like in Costa Rica, it is about 1/3 of the banana. So that's an immense amount of money if you take the entire area into consideration. Now, if you look at a banana crop, in a wheat crop, I showed you, and so in medieval times, the wheat crop was about our size. Nowadays, contemporary wheats are much shorter, but if you talk about banana, you're really standing in the crop. There's a lot of shade. And here you see a banana variety that was produced by Rahan in Israel that is way shorter. But this improvement, this is all both Cavendish, this improvement is only done by mutation breeding. So far, new varieties that come to the market are usually through mutation breeding. Thus far, classical breeding programs, are just a handful around the world, have not been able to develop varieties that can compete with Cavendish. But part of that, of course, is also due to the very limited know-how of the genetics of banana. Now sequences are being generated, breeding is being accelerated and started using all the genetic tools we have, so that is promising. And of course, we can use and really deploy the immense diversity that is available, but that is really untapped so far. So this really has to change. And that's what we are currently doing. So we are setting up a collaborative effort with Chiquita and Kijin and Muserradix, one of the spin-off companies of Wageningen University, to start a classical breeding program, but driven by modern tools to do gene discovery and really improve the throughput and quality of breeding. Now, if you talk about gene discovery in the fungus, we have now many sequences generated. So this is a slide from the paper of Heini Widu Nugar from Martijn Reb's group in Amsterdam. And here they have discovered or described the gene 61A, which really contributes to virulence if you knock it out. So here you see the water control, the wild type. If you knock out the gene, you have a reduced infection of the plant, as you can see also here. And so the identification of genes involved in pathogenicity, and of course we looked into 6 effectors because they are so important in the Fusarium oxysporum tomato complex or melon, and of course 6 genes in the Fusarium strains causing Fusarium wilt in banana, also very important, but of course they're not necessarily the only effectors, so that's where we have to keep an eye on. So another thing we really need is we need really a way higher throughput for screening varieties. I didn't mention that, but you know, wheat we usually inoculate when it's 10 days old, but banana you have to wait for 2 or 3 months before you can inoculate the plant, and then again you have to wait for 7 or 8 weeks before you can score the plant. So that is really slowing us down tremendously. Fortunately, we are involved now in a project driven by IATA and funded by the Bill and Melinda Gates Foundation. So we have 2 vacancies for 2 PhDs, one on bioinformatics to work with me and with Michael Seidel and myself, and one on the effector biology together with Carolina and Juliana. So if there is anybody who has an interest, send me an email, contact us, because those vacancies will be opened very soon. Very much focused on developing tools to improve the throughput and precision of phenotyping for resistance to Fusarium wilt in banana. And with that, I would like really to thank the team involved in what I presented today in our Fusarium wilt effort. I appreciate the collaboration with Eva Stukenbrock on the genomics and analysis of the phylogenomics of the Fusarium complex in banana. I also would like to show you the book series Andre Brandt and myself are working on achieving sustainable cultivation of banana. Volume 1 is published, Volume 2 will be published this year, and we're working on setting up Volume 3, focusing on plant diseases for next year. So the book of this year is on genetics and breeding. And with that, I would like to thank our sponsors and collaborators, everybody who was involved in making and enabling us to do this research. And finally, I really would like to thank the fantastic team at the Laboratory of Phytopathology in Wageningen University. Thank you for your attention.
Thank you very much, Gert. Very exciting talk. I will hand over to Lisa, who will handle the question for the remaining of this session. Thank you.
Thank you very much, Prof. Kema. So we have our first question. Wheat research is well funded by private and public resources. Banana, not so much, or it seems. How do we change this?
Yeah, I mean, I think one of the reasons to change it really is to swamp the area with information. So since we started working on banana, I have been surprised myself how easy it actually was to attract funding for banana research compared to wheat research. You know, of course, the Netherlands is a small country. Our wheat crop is about 100,000 hectares. And of course, it is more and more increasingly difficult to find funding for research for wheat in the Netherlands. But banana has such an immense appeal to many people that it was way, way easier to attract funding for banana, particularly because it's also a crop that is really surrounded by social issues. As I mentioned, there are millions of smallholders involved. You know, many people have employment in the banana sector. And it is a very important staple food. You know, of course, not as important as wheat, but in many developing countries, banana is an absolute staple. And yet there is these immense disease problems. So, you know, telling the story is not that difficult. It's very compelling and very easy to understand that we need to do something. So we have attracted really funding from many companies, but also public funding from the Netherlands and elsewhere. And we are still working on big programs to make sure that we also contribute to food security, because, as I said, banana is an important food crop.
Thank you very much. Next question. First, a compliment from an attendee. Thanks for the great talk. We have promising studies on the soil microbiome engineering to control Fusarium wilt, i.e. the use of beneficial and antifungal microbes as probiotics to induce resistance and inhibit Fusarium growth. Is something like this possible and sustainable?
It's definitely possible. I don't know how sustainable it is, but that's why we have to do the research. So the advantage, of course, now is that we have a fantastic tools to study this. So we're also setting up experiments and projects to do this. So there are very, very interesting underlying questions. As I mentioned already, TR4 is super pathogenic on banana, but it can grow as an endophyte in another host. So that is raising an immediate, very interesting question. But of course, the effect of the microbiome on the expression of resistance genes is another area that is essentially black. So yeah, I think it's a great opportunity. We really have to do it. On the other side, that's why I also show the book of Saluri. 100 years ago, they already used beneficial microbes in all kinds of different settings, and yet it did not help to maintain Gros Michel production. And so, and maybe I'm biased because I was trained as a plant breeder, but if you have a susceptible crop, you're never going to save that crop with all kinds of other methods. We have to then work also on developing new varieties and really deploy the resistance that is available. But of course, before that is ending up in new varieties, that takes us another 10 years or so. So meanwhile, we have to deal with the situation. And yet, so number one, it's a very interesting area, and it definitely needs to be done.
Another question, does the Fusarium subtropical race 4 cluster with the tropical race 4?
No, it doesn't. It's a different, the subtropical race 4 is essentially a race 1 strain. And so these are strains that can infect or can cause disease, or at least generate symptoms under abiotic stress. So we have also shown that in the greenhouse. So if you reduce, for instance, the pH, a race 1 strain is causing foliar effects in Cavendish bananas. If you raise the pH to 6, the effect is gone. But thanks for the question. So subtropical race 4 is not in the TR4 cluster. It's a different species.
Thank you. So is there any correlation with tropical race 4 infection severity with the age of the plant? So does the fungus prefer young saplings in the field?
It does. It does. Younger plants are usually very, very sensitive to the fungus. So that's why when we receive tissue culture plants, right, so they are shipped to us by our collaborators in small plastic boxes. And so we first harden the plants for 2 to 3 weeks, then we grow them to 2 to 3 months, also to make sure that our phenotyping in the greenhouse is relevant for the field conditions. Of course, since we do not have that many genetic data, I'm not excluding that there may be genes in banana that are expressed primarily in the adult plant stage or primarily in the, let's say, younger age of the plant, like we see in wheat crops. In wheat crops, it's very well known we have adult plant genes. They are only expressed while the plant is growing at a later stage. And in banana, we just don't know that. But what we have seen from the correlations we ran so far, the greenhouse screening is very relevant and predicting the in-field susceptibility or resistance of the crop.
Thank you. So we have many questions. I'm just going to try and combine some of them that potentially can be. There is a question here about the QTL. You haven't shown all the chromosomes after mapping the resistance QTL to race 1 on chromosome 10. So did you expect a single dominant gene?
Well, we didn't know what to expect, to be honest. Yeah, but we were, let's say, pleasantly surprised that we actually mapped one major QTL. I cannot give you too many details because the paper is under review right now. But it is definitely not impossible that the resistance of Cavendish to race 1 is driven by a single gene. And that's why I also gave the example of the gene that was cloned by James and put into Cavendish that gives full protection, right? Now, the way how to deal with this, of course, is that whenever we have identified and validated that gene, we see, do we see a copy of that gene in Cavendish and knock it out and see whether that turns Cavendish into susceptible to a race 1 strain. So that's what I can say at this stage.
Thank you. Another question. To what level is the level of resistance with the race 1 on Cavendish? So at what stage is the fungus blocked?
Oh, prior to the infestation of the corm. So it's really maybe right away after entering the root system.
All right, thank you. So a question about the rate of breeding. Is there any way that we can speed up banana breeding, extending the photoperiods or hormonal treatments?
Yeah, sure. So for us, that's why I mentioned the initiative. For us, speeding up breeding is really using the tools that are, let's say, common sense and that are used throughout the breeding of many, many other crops. Until very recently, these tools were not available for banana breeding. Fortunately, due to different programs, so one of them is a program funded by the Gates Foundation that was already running in East Africa at the ITA. Many tools have been developed, but of course at this stage there is not a private breeding program in banana. That is very, very different from many, many other crops. So that's where we saw an opportunity, and I think if you have a very focused drive and develop the right tools, and that's why we're happy to work with Kijin, who has been very instrumental in developing technology to speed up breeding. We are convinced that we really can push breeding in banana way faster than it used to go.
Thank you. So what is your take on the distribution of GC-TCV soma clones developed by TBRI? Do you see them being part of the long-term solution or not? And maybe you could comment on the stability of induced resistance traits to the genetic material across the clonal propagational cycles.
Yes, thank you. That's an interesting question because to our surprise, I have to say we have worked with the company on mutation breeding and see whether we can use that to improve, or let's put it this way, to reduce the level of susceptibility to TR4 in banana. And to my surprise, and I think to the surprise of everybody who was involved, yes, we were able to select plants from that mutant population that had a very significant level of resistance to TR4 that is currently being tested in the field at various locations. So yes, there is an angle really to see, can we improve Cavendish for resistance to TR4 by mutation breeding? I have to say that approach, and that's the other side, that is kind of entirely focused on Panama disease. We haven't seen any improvement of resistance to black Sigatoka following that approach. So that's a big question mark. And so principally, yes, possible. Practically, I think this is not the way to go because we still maintain Cavendish as a monoculture around the world, super susceptible to black Sigatoka. And so we have to do at least both. We have to breed, we have to diversify the crop because that makes the crop much, much more sustainable. And that's what we have to achieve. On the GC-TCV clones, we have seen them in the field. We have tested them to TR4. I usually say those clones are not resistant. They are at most less susceptible. And of course, a less susceptible variety can never be the solution, neither be a sustainable solution for the current issues on TR4 in Cavendish.
Thank you. So spinning off of that question, to what extent is the coming up of this disease related to large-scale monoculture of one variety of species? And what do we learn from this in designing future sustainable food systems and potentially using something like the promising transgenic bananas with TR4 resistance?
Sure, yeah. Well, that's what I tried to say to show in my talk, right? So, I mean, Cavendish was really coming to the picture because of the demise of the Gros Michel-based production in Central America. And at that stage, there was hardly anything to replace Cavendish. And fortunately, they found Cavendish, which had an edible fruit. Okay, the quality was maybe not as good as Gros Michel, but now we see it's globally appreciated, at least in Western markets. And it turned out to be super resistant to race 1. And that, of course, boomed the Cavendish production up to the global monoculture that we're dealing with now. And of course, in that way, in this whole movement, Cavendish essentially became the vehicle of spreading TR4 because it's everywhere, and it's very easy to jump from crop to crop for TR4 to really disseminate in regions, and that's what we see. That's what we see in the Mekong area. That's what we see in India. I'm afraid we're going to see that also in Africa. And that's what we see now. It jumps into Latin America. And the only way to eventually deal with this is diversify the crop. And, you know, just let's look outside our borders. I come back to wheat again. We have hundreds of wheat varieties around the world through a period of 100 years, a century of wheat breeding. And that has helped us a lot to deal with diseases, period. This is all non-heard of in current banana production. And so we don't have anything else to replace Cavendish. So that really puts the pressure, really a lot of pressure to come up with solutions. Yes, to deal with it now, but also to prepare for a way more sustainable production system in the future. And transgenics, can they be part of that? Why not? I mean, I'm not objecting to transgenics at all, but where we have to deal with is the practical issues, right? So we cannot grow transgenics in any or in every country. So whenever that legislation makes it available, bingo, we have genes identified, we already have transgenic resistant varieties. Of course, we haven't touched upon genome editing, which is a fantastic potential resource, but we should not forget one thing. Genome editing stands or falls with the number of genes that we have identified. And that's why I gave you the comparison with wheat, right? In banana, we have only a few genes. So if you don't have targets, genome editing doesn't help you a lot. And so we have really to do all that basic genetic work to identify and validate genes that can be used to improve the crop.
So we've now identified that there's a need for more genetics and that we need to look at how this can be implemented in a manner that we aren't just sitting and waiting, but we're doing something about it while we prepare potentially new clones. What are the current practices that are being done on field conditions or field level for these, for controlling tropical race 4 with smallholder producers and at a commercial level?
Yeah, well, let's start with the last one. At a commercial level, we are working with several companies that develop fungicides to control Panama disease. I haven't addressed that in the talk, but fungicides would be most welcome to help us to control Fusarium wilt in banana. The practical issue, though, is the fungus is very, very sensitive. And so the dogma that existed for quite some time that you cannot control this disease with fungicides, that's not correct. The fungus, this whole diversity that we have identified, is very sensitive to fungicides, the regular fungicides, you know, because they've never been exposed to fungicides. So far, no resistance issues. The problem is, how do you get a fungicide in the soil to reach the inoculum? That's a major practical thing that is currently being addressed as well. And so, yes, so fungicides are on their way and they would be very welcome to deal with the disease. I'm not an advocate of fungicides, but if you don't have anything, you know, it's a very valuable contribution. And black Sigatoka, of course, is showing that very well. That's number one. Of course, there are also other ways. I showed inundation. There are also now smarter ways to, you know, to disinfect soils, even in biological ways. So we also have experimented that in the Philippines with, which was successful. So we can kind of sanitize soils, but of course there is always the cost picture there, right? So, you know, we have to be very inventive how to deal with the disease. Something I did not show in the talk is we have grown bananas in the greenhouse. But the principal thought about it was, why don't we lift the crop from the soil? So we grew bananas in substrate. And they produce fruits, they produce bunches of 40K. So if you want to avoid the problem, you also can lift the crop from its problems. And so we have to be very pragmatic, we have to be very creative, and I'm sure there are various ways how to deal with the disease. The major thing, though, is we have to prevent that it's entering new zones. And so biosecurity, sealing off, isolating infested areas, do all the checks, and that's why I show the area monitoring is really, you know, to prevent that the disease comes in, and when it is there, implement all the things you need to do to isolate that zone. Australia is a key example of that. you know, there are 2 incursions in the prime production area in eastern or northeastern Australia, and they really isolated that farms, literally. And that did not prevent secondary infections, but it really reduced it a lot.
Thank you very much. So I'm going to take one more question because
Okay.
I think everyone's time is also sensitive, and I don't want to lose your interest in the talk. So while we were talking about the monitoring at a regional level through the aeroplanes, does this app or this tool differentiate between black sigatoka and nematode damage by any chance?
That's too early to say at this stage, but you know, we are using you know, the area monitoring is really a matter of artificial intelligence. It is really machine learning, combining data sets that are available in many production areas, but are not, well, they are, number one, they are not disclosed. And secondly, there's usually an enormous amount of data that is unexplored. And so if you combine those data sets, you may be able to differentiate for sure plants that are suffering from healthy plants. And so that's why I said, you know, proactively you have to identify risk zones and sample those in particular. You cannot sample 40,000 hectares. You really, in a way, have to identify what are areas that look different, and how can we use that technology to zoom in on those areas to prevent, number one, incursions, and secondly, dissemination.
Well, thank you very much, Prof. Kema, for the very fascinating talk. I'm going to redirect all the questions that haven't been answered to the Slack channel. I can also email them to you. So
Okay.
we will try and wrap that up in the blog. I think that'll be a good platform to finish this very hot topic. I'm very glad for all the participants that could attend. So thank you for making the time to be with us today, and to the viewers for participating. I just want to let you all know that our next presentation will be on the 4th of May, and it'll be Dr. Sally Malawa, and she will be looking at technology-driven cassava pest management. So thank you everyone for your time today. Thank you very much, Dr. Gert Kema. Have a wonderful day further.
Likewise, you're welcome. Bye-bye.
Goodbye.
Bye-bye.